I think we can start now. Okay. I prefer to off my camera to maintain bandwidth of Wi-Fi. Okay. Thank you.
I think TIC sir haven't joined yet, right?
TIC sir joined.
I'm already joined.
Okay, so I request you to keep some to say some few words and kindly address the participants. So.
Okay, thank you, Dr. Tamang. A very good afternoon to all. I, on behalf of Shantipur College, welcome you all to join the international webinar on the topic Recent Trends in Biological Science. I appreciate the sincere involvement and proficient engagement of the HOD of Botany, Ms. Swapna Tamang, convener of the webinar, Srimati Sulagna Roy, and all the faculties of the department. My special thanks goes to Dr. Dipankar Bhattacharya, convener of IQAC, and Dr. Anirban Bhattacharya, convener of the seminar subcommittee, for assisting, cooperating, and stepping forward to any academic events of our college. I hope the lectures delivered by our eminent speaker, Dr. Roy and Dr. Ghosh, will highly benefit the students and other participants attending the webinar. I heartily wish the webinar a remarkable success. Finally, I extend my heartfelt thanks to all the participants and the respected distinguished dignitaries attending the webinar. Thank you.
Thank you so much, sir. So, good afternoon to everyone present here. Warm greeting from the Department of Botany, Shantipur College. I'm Swapna Tamang, Head of Department of Botany, Shantipur College, and it gives me immense pleasure to welcome you all to the one-day international webinar on recent trends on biological sciences, organized by the Department of Botany. We are truly delighted to host the session where academic minds come together to explore and progress to possibilities to the field of biological sciences. I'm honored to extend a warm and respectful welcome to our distinguished speaker, Dr. Chitran Roy, postdoctoral researcher at Pennsylvania State University, USA, and Dr. Dipayan Ghosh, postdoctoral researcher associate at the Center for Rural Development and Technology, IIT Delhi. We feel privileged to have you both with us, and we look forward to gaining insight from your expertise. I also take the opportunity to warmly welcome our TIC, Dr. Subrata Roy sir, IQAC coordinator, I think is not present, Dr. Dipankar Bhattacharya sir, and the convener of this seminar organizing committee, Dr. Anirban Bhattacharya.
No, I am present.
Okay, thank you, sir. Thank you. We have with us our convener, Mrs. Sulagna Roy, co-convener Sangati Kundu, and Rajkumar Sharma of Department of Botany, who have worked together to make this event possible. I also extend a warm welcome to our enthusiastic participant, student, researcher, and a faculty member from various institutions for joining in this academic exchange. Today's webinar aims to shed light in some of the most relevant and emerging trends in biological sciences, a field that is constantly evolving and offering new insights into life, sustainability, and innovation. We hope this platform will encourage young minds and inspire further exploration. With that, I once again welcome you all now and invite everyone to engage wholeheartedly in today's session. I request our convener, Mrs. Sulagna Roy, to proceed with the session. Thank you.
Thank you, madam, and good afternoon to everyone. It's a great honor to welcome you all at the first online international webinar which is jointly organized by Department of Botany and International Quality Assurance at Shantipur College. And this webinar has been organized with the aim of fostering academic exchange, encouraging new ideas, discussing emerging developments in biological research. And we hope the presentation and interaction today will spark meaningful and collaborative thinking also. And on behalf of organizing team and a convener, I again extend a warm welcome to our esteemed speakers, respected patrons, faculty members, research scholars, students from across the globe who have joined us today. And I also convey my heartfelt thanks to respected patron Dr. Brojo Kishore Goswami, President of Governing Body, and Dr. Subrata Roy, Teacher in charge, Shantipur College, for their constant support. Okay, now I preferably off my camera. Okay, and it's my privilege to introduce our first plenary speaker, Dr. Chitran Rai, postdoctoral research at Pennsylvania State University, USA. He will be presenting on the topic a new dimension of phycobilisome and phycocyanin in medical science, emerging therapeutic applications. And with a great respect, I now invite our first esteemed speaker to commence their presentation. We look forward to gaining valuable insights from your expertise. And Dr. Roy, the platform is now yours.
Hello.
Am I audible, sir?
Yeah. Am I audible?
Sir.
Okay, thank you. And it's my great pleasure to talk here in front of you, my senior scientist and my upcoming researcher and the scientist also. So before that, I would like to share my presentation. So it's visible.
Yes, sir.
The PowerPoint is visible.
Yes, sir. Visible.
Okay. So sorry. So first, let me introduce to myself. Before after completing my PhD, I went in the Israel, and after that, I joined in the Pennsylvania State University. The work on the phycobilism and the phycocyanin, I work basically in the TechMed, Israel Institute of Technology. Same there, I purified the photosynthetic component, specifically in the phycobilism and the phycocyanin, and then process further. But this is really in the upcoming scientists. I'm giving the presenting the upcoming scientists in front of the upcoming scientists. So I just simplified my work from the beginning to end. So first, I would like to say the light energy in the visible light, the photosynthetic plant, they can absorb only the visible light, and they can activate their chlorophyll energy. So in the range of 400 to 700, they actually absorb the light. But in the case of the red and the blue light, they are more activated and continue their photosynthetic activity. In the basically red and the blue and the apart from the green light, because the green light they can imitate the each wavelength of light, and apart from the green light, all other light become absorbed, and only the basis of the red and blue light they are taking and activated. In the graphical represents is so the most plant have the leaf that is reflect the green light and absorb the other color, and energy in the red and the blue light most stimulate the photosynthesis. Next step, I would like to say the photosynthesis done by phytoplankton, kelp, and the photosynthetic plant, different kinds of cyanobacteria, nutrient, they all do the photosynthetic. And the photosynthetic is basically the endergonic chemical reaction because it's required the energy, and it accepts the sunlight, the water, and the carbon dioxide, and emits sugar and the oxygens. And next step is that is the some microscopic structure of the plant guard cell, where it's showing the carbon dioxide, they intake the carbon dioxide in and the oxygen out. Yes. So now this is the chloroplast, where photosynthesis is doing in the chloroplast. In the chloroplast, when you cross section of the chloroplast, the thylakoid membrane, where the all all the protein of the chlorophyll molecule embedded in the thylakoid membranes, and the photosynthesis is the light, as it is a light dependent reaction. So all reaction is located in the thylakoid membrane, and after that, this is the I just showing the elaborate structure of the all proteins, where it's showing the photosystem 2, they can activate due to the presence of the sunlight, and the water molecule become breakdowns, and it can take the electron. This electrons is goes to the photosystem 2. The electron then passes through the photosystem 1, then activate the whole things. The protons and molecules are come out through the ATP synthase molecule, proton move, and the ATP and NADPH is produced. In here, it's showing the light dependent reaction in here. That is when the amount of the proton molecule become more in the inside the cell, it can accelerate the energy, and the ATP synthase is become activated. In that cases, so ATP will be used later, production of the ATP, and the NADPH is used later. That is the some graphical representation which is showing the after the activation of the sun, there is the photosynthesis 2, and when it can capture the light energy, and the continue of the protein is the photon is the activated in the chlorophyll molecule, electron become excited, and in the same time when the excited electron become passes through the phaeophytin, and this electron carrier of the protein, and each electron again filled the vacuum condition of the chlorophyll molecule, and the excited electron can passes through the cytochrome P6F through the electron transport chain in the presence of the phaeophytin and the other molecules. There's some graphical representation in here. So when it comes in the cytochrome complex, the port through this port component, proton can come inside the cell from the outside of the lumen conditions, and then electron can passes to the photosystem 1. The photosystem 1 chlorophyll molecules become activated. Each electron, and when is activated through the sunlight, and each electron goes to the A1, A0, A1, and the FES system and the FD system, and it's the production of the NADP to NADPH. And this when the amount of this proton molecule in the thylakoid lumens, it's in that condition ATP synthase become activated. That means when the proton content is the lumen become more, and this is passes through the ATP channel, ATP synthase, and during the ATP synthase is motor can be rotated, and it's convert the ADP to ATP, formation in the thylakoid stoma. Each ATP and NADPH is used in the further dark reactions. So now the photosynthesis is occurred in the higher plant, also in the blue-green algae. The architecture of the cyanobacteria is a little bit different with the plant architecture, but the photosynthetic system is the same, because in cyanobacteria they also have the photosystem 1, cytochrome B6F, and the photosystem 2, and through these channels they are become activated and produce oxygen and NADH. In the basis of but how the cyanobacterial become activated photosynthetic compound, because in case of that's the pH2, in the above portion of the pH2 is contain the different type of proteins. It's contain the allophycocyanin, phycocyanin, and the phycoerythrin. So based on their chromophore group, they absorb different kind of wavelength. And the whole complex, that is the component of the whole complex, that's in here we can denote it in the circle. That's a whole complex, that is allophycocyanin, phycocyanin, and the phycoerythrin is called the phycobilisome. It absorbs the light between the 660 to 670 nanometers. So now in my postdoc, so I cultured the cyanobacterial and harvest the cells and is grow the cell. It's in here, it's showing the very beautiful color of the cyanobacteria as well as the green algae. So after culturing or harvesting, and we can purify the phycobilisome and the phycocyanin proteins. So in here now my work was to purify the protein and the structural determinations. So in that cases, I particularly purify the phycocyanin. That's a component, and a phycocyanin is a two subunit, has a two subunit, alpha and the beta subunit. After purification of the phycocyanin proteins, I was trying to crystallize these proteins in the different crystallized screen or the different crystallization buffer. So first step, I got the very tiny crystal which are reflecting in the showing with the blue color. These are protein crystals, and after optimization, so I got a very big crystal size, and that crystal we were the pack, and we sent the crystal in the France for the data collections of that was the European EFRS. That's the European Synchrotron Research Facilities Lab. So I got the crystal diffraction data from them, and I determine the structure in the resolution of the 3 angstroms. As the resolution of the 3 angstroms are not quite good data enough, so I did not found any chromophore group bound on the protein crystal. So, however, I got and define the structure which we can electronically that's a blue structure is show the electron density plus density value is the number is 1.7. I got the protein crystals. So that is the phycocyanin protein crystal which we define the structure of the phycocyanin or the cyanobacteria. Next step, I mean our team is also purify the whole complex of the photosynthetic, light harvesting complex that's called the phycobilisome. But I purify the phycobilisome complex to the sucrose density gradient centrifugation, and keep the sample for the next step for the data collections. And after that, simultaneously another lab also doing the same kind of work, but due to the due to the Hamas attack in the Israel, so we all come back to the India, so we cannot go to the further. So, however, in the next another lab are also doing the same kind of work, and they also they also did the whole phytobilism complex structure. This is the prion density structure and particle structure, and this they actually solve the structure between the 2.7 to 3.5 Armstrong resolution, and that is the light harvesting complex of the blue green algae. They actually purify. Another group of the Chinese group, they all actually complete that project. So, however, so that is the scenario of the structure of the phycocyanin and phycobilism protein complex. And but the thing is the application of such kind of work, because that is not a valid. Without application, the science is not a valuable. So what kind of application is that? So in case of blue green algae, there's a different kind of blue green algae, that is the spirulina or the cyanobacteria. In case of the C phycocyanin, that's the phycocyanin, or the phyco is a part of the phycobilism complex, is a one kind of phycobilic protein. That's a chromosome found in the blue green algae, such as the spirulina. This often is the dietary nutrients supply or the variety of the pharmacological properties it applied in. Cyanobacterial also contain the different colored protein, which colored compound is really used, applied for the cosmetic world. So extractions of the phycocyanin or phycobilism, basically the chlorophyll group, they actually use for the cosmetic industry and etc. So that is the chromophore group, chromophore structure. So based on the chromophore conformation, so they actually absorb the light in the different wavelengths. So extraction is a very challenging of these the phycobilin protein. So extraction and purification, it's also act as antioxidants, anticoagulation, and prevention of the DNA damage activity. It's already being established in the that is the spirulina based culture, because this is in the recent nowadays actually spirulina act as a scavenger activity. It has a scavenger activity, is a nutritional value. It's also formed in the powder and the tablet formulated. In India, basically the NB laboratory, they also obtain the spirulina proteins and marketize the tablet and the dust form. So that is the some online available some spirulina protein and the tablet. However, so now the question, how spirulina help during the chemotherapy? The question is yes, the spirulina can also it's already been published. It's also in the Translational Cancer Research Institute. They already use the spirulina for the cancer treatment. Apart from the cancer, they also maintain the blood cell levels, boost the immunity, and reduce the side effect of the other contaminated cell in our body. So lastly, I would like to be showing some graphical picture where we can see the plastocyanin. They act on some different kind of cell checkpoint, and it also helps to cell apoptosis or the cell autophagy, activated our immune system, and some cancerous cell checkpoint, they can also activate the P338 or ERK pathway. So it can also helpful to treat the cancerous cell and also that actually. So now I just want to finish my talk, and this is very much thankful to all of you, because it's a great pleasure to me to be front and discuss a little bit scientific valuable opinion in front of you. And thank you. Any question?
Thank you, Dr. Roy, for your enlightening presentation on therapeutic potential of phycocyanins. And it's really a very eye-opening presentation. Your work truly opens new horizon for integrative medicine and biopharmaceuticals. Our question sessions are now according to schedule. It's after the second speaker.
Okay, thank you, Dr. Roy, for your insightful
And
presentation.
now,
And so now I can.
sorry, preferably. Hello. Hello, am I audible?
Yes, yes.
Okay. I request kindly the respected IQAC coordinator, sir. I kindly request you to say a few words in this webinar. Please, sir, share something with us. We'd be grateful to hear from you.
Yes, sir.
Okay, actually, since it is already started, so I think there is no meaning to say something in between it. So just continue the process and so we'll start the next speaker. It will break the sequence. Okay, so it is no matter that whether I say something or not, because since you are all part of this this college, and we definitely look forward to this enriching and inspiring session. So just continue it. Okay, and I think it is better if you take this question answer session now, because if some participant has some questions, then they can ask now. Otherwise, they need to keep it in mind, and then after the second session, they ask questions. So it is up to you actually. It is just my suggestion. Okay, let's continue.
Okay, sir. Thank you. And definitely, now we now open the floor for questions. Then, please, students, participants, please feel free to drop your questions in chat box or raise hand features used. We'll try to address as many as the time allows.
So I have a naive question. Actually, you know that I am not from this field. I'm from physics background. But my question is very simple: that what is the application other than this cancer and other things? There is what is the application? And just my curiosity is how this experiment is performed. If yes.
Yes, the experiment is because science is it's depend upon the three factors. There is why we are doing the science and what's the applications, and to know the fundamental queries. In here, the photosynthesis is one kind of phenomena, and nowadays the researchers of scientists, they are actually applied their phenomena. I mean, natural work they actually applied in the medicinal field or in the other industry also. When I was in the Israel, the other group of the scientists, they actually use the photosynthetic phenomena, and they actually developing the artificial photosynthesis for the fuel productions. There's the difference. In the medicinal field, because as that is the phycocyanin or the phycoglycogen, they have the scavenger activity, because they acquire some free radical of oxygen. So in that cases, they try to apply the cancerous cells in the different kind of checkpoint. So they are trying to apply such kind of use such kind of chlorophore group or phycocyanin protein, and applied to enhance the pathway where the cancerous cell become reduced. So that is the application actually. This is not a direct application you can't say, but it can enhance the immune system or boost the immunity to shut down the cancerous cells or other notorious cells also.
Okay. Thank you, Dr. Rai, for a nice explanation.
We encourage all students to come forward with their queries or reflections. Please feel free to unmute yourself or use the chat box for comments or questions.
I can see already one question in the chat.
From Shubir. Okay, sir, I will read for you. The question is, have any clinical trials has been conducted using phycocyanin or phycobilisomes, derived compounds for therapeutic purposes?
Right now, that this system is only the trial system, because many scientists they are actually using different kind of cell line and treated the direct in the solution of the phycocyanin or the chromophore group of the phycocyanins, and they are already checking. So based on their data, based on their in vitro data, they are just think that yes, the phycocyanin and the phycobilism component is a make in the near future is a can help to reduce any other the cancer cell or any other act as an antioxidant, etc.
Okay. Thank you, sir. Thank you very much. Thank you. Any other questions? Please feel free to unmute to all participants and students. Oh, there's another one. Konkona Ghosh said there, what are the known what is the pharmacokinetics and pharmacodynamics of phycocyanin in the human body?
Yes, sir, that is the answer may be the same which I answered before, but again, just I would like to say that is not a direct phycocyanin derived protein or the phycocyanin or phycoprotein in the human body, but it's all applied in the cell culture. And the scientists they are also doing what kind of pathway or what kind of how actually they work as after treatment of the phycocyanin, how actually they behave. So they are just searching, and the many cases they get a very convinced result. Many people they first in the Israel in my site lab they also doing such kind they are also doing such kind of work, but their paper is still not published, and I think they will publish soon very early. So thank you.
Thank you, sir. Thank you very much. Any questions for participants? Okay. Sir, I have a question shortly. Like, if
Sure.
spirulina platensis, like we mentioned earlier, that spirulina platensis dry powder, and there is any significant observations on this microalgae?
There's not a significant extent, but is already the edible. So you can also take the tablet or the because deep spirulina protein is also it has the activity in the scavenger activity. So when each protein, it's also reduce the free radical or some protein. Those are the very anti-aging activity because aging is depends on the free radical formation. Huge amount of free radical formation in the body, they can immediately going towards the aging. So each proteins have the capacity to capture the free oxygen. So that is also help to reduce and simultaneously they boost the immune system. So I think this is actually the application of the spirulina, and many scientists they use this nowadays.
Okay. Okay, sir. Thank you very much. Thank you for your explanation. Thank you. Okay. Now let us move to.
I think there is two more questions. Please check it.
Oh, acha. It's later. Right. Very sorry. Very sorry. Sir, I will read it for you. There's a Ashmat Parveen. Are there any development in increasing synthetic or recombinant phycocyanin with enhanced medical properties?
In case of recombinantly, definitely this in here we culture the from the cell, but recombinantly we can or we may design the protein, and we can also help to the medical science. Maybe it's possible, but the main thing is the chromophore group, the bound chromophore group, because chromophore group is very much important, phycobilins that is called. So if the recombinant protein have bound with the chromophore group, I think it's not possible to in the UP applications in the medical science.
Okay, sir, there's another one. Is there any toxicity concern or co-indicator with long-term use for phycocyanin in medical doses?
Yes, definitely. There are some that if beyond the threshold level, if you intake any kind of thing, is become toxic. So in case of phycocyanin, if you so as per the doctor prescription or the doctor recommendation, and this is toxicity is not directly proven, but as per the doctor concern or other scientist concern, they can given the dose dependent activities. So definitely. So before that, if you take it beyond threshold levels, it may concern to the toxic level.
Okay, sir. Thank you very much. Thank you for your question explanation.
Thank you.
I further request to all candidates or participants, do you have any other question? Please feel free to unmute. Okay, this is another question from Shorab Roy. Were there any major challenges in obtaining pure phycobilism crystals considering their complete structure?
The pure phycobilism, if you would like to structure of the phycobilism, so, I mean pure phycobilism, it's a little bit tough because it's a big structure. So if you, but it's a very challenging. So that's why we people were doing the cryo-electron microscope. But if you'd like to crystallize the big protein, it's a really challenge.
Okay, thank you. Any other questions from anyone? Participants, please feel free to drop your questions. There is another one. How might phycocyanin be tolerated to treat inflammatory disorders? So, right?
Yes.
Uh-huh.
As this is the some natural product, so I have already told there is already concerned in the physicians or the doctors. So when you take the phycocyanin for the information, so it's also depend on the physician and the doctor concerns. So based on their suggestion, how much you will take the phycocyanin or how much is the inflammatory disorder. This depend upon their actual physicians.
Okay, sir. Okay, sir. Thank you. Now, there's another question from Labidul Rana. Why phycocyanin considered potential candidates for drug development?
Yes, because first of thing that is this is the drug which we develop from the phycocyanin. It's the least harmful because there is a bioproduct based phycocyanin, and as if we properly if we treatment properly in the presence of the phycocyanin protein, it can also activate our immune system, and it in the after digestion of the ingestion of the body, it can easily pass out from our body after their work, because it's according to my perception, it's really convenient to use the chemically synthetic product rather than the phycocyanin, because it's already the bioproduct, so it may help or the may less effective to your human body system. These are my perceptions.
Okay, okay, sir. The next question is, are there any side effect or risk associated with using phycocyanin in human treatment?
Yes, I have already told before, because this is already so for taking the phycocyanin or any plant-based products, after discussion with the physician or doctors, so they will suggest how much it will take, how much it's help to your body system, because each and everybody after taking the any other kind of drug, their repercussion is different to the different body immune system. So based on the physician or based on the doctor, they will suggest how much phycocyanin is related to your body, or the how much spirit will take, tablet you will take, which is good for your body system. So based on the physician or doctor, uh, they will suggest to which you qualify best.
Okay, sir. Thank you. Next question, probably it's the last. Future prospect of phycocyanin in medical science.
Definitely, because that is the phycocyanin is the basically chromophore found contains. So that to know the exact structure of the and definitely there is under development of the process, and as the phycocyanin is the plant bioproduct, so it will help in the medical science or treatment of the any artificial treatment of the as a drug. So if the scientist can establish this the phycocyanin uh, treatment, so we can ingest the bioproduct rather than the chemically synthesized product. So definitely that is a effect of the phycocyanin or the plant or the poultry algae based product. Thank you.
Thank you, sir. Thank you very much. Okay, all participants. Now, to let us move on to our second distinguished speaker, Dr. Dipayan Ghosh, Postdoctoral Research Associate, Environmental Biotechnology Lab, Center for Rural Development and Technology, Indian Institute of Technology, Delhi. He will speak on the topic, Cannabis Beyond Controversy. It's a scientific advances in cannabinoid research and breeding with an Indian perspective. Okay, Dr. Ghosh, am I audible?
Yes, yes, yes.
Okay. Now, I now invite our second esteemed speaker to commence their presentation. And Dr. Ghosh, the platform is now yours. Thank you.
Is slide visible?
No, Dr. Ghosh, till now.
Now?
I think now it's visible.
Thank you, ma'am.
Thank
Good
you.
afternoon to all of you. Respected teacher in charge, head of the botany department, IQ coordinator, and all the respected dear participant and respected organizing team. So today I am talking about my PhD work. I just started in 2018. The topic is cannabis beyond controversy, scientific evidences, scientific advances in cannabinoid research. Is it visible? If not, slides.
Sir, slides are clearly visible now.
Okay. The topic is cannabis beyond controversy, scientific advances in cannabinoid research, reading with an Indian perspective. So before start, cannabis belongs to the family Cannabaceae. It is an annual herbaceous plant, commonly known as Indian hemp. In literature, there is three different species of cannabis was reported. The first one is Cannabis sativa, reported by the Linnaeus in his book Species Plantarum. Another one is Cannabis indica by Lammer. And the third one is Cannabis ruderalis through Russian taxonomist Janis Chusky. But recent taxonomic research considered the genus Cannabis.
Sorry to interrupt. We will start on your first slide. The slides are not moving now.
Just 1 minute.
Okay.
Is it visible now? Change.
It is visible, but it is stuck in the first slide, not moving. Now
Now it's okay.
make it full screen.
Okay. Now?
No.
From my side, it is in slide mode. Now is it visible or full screen?
It is at least in my laptop. It is visible, but just the introduction, but not in full screen. The slides is introduction, but not.
Sulabna ma'am, can you help me?
Definitely. It's totally visible, but not in a full screen. I tried to fix it on a full screen. I think these slide settings are only visible in a full screen from your device, sir.
I keep my slide in presentation mode.
Your presentation is totally visible. It's just perfect, but just not in a full screen.
Okay, I can.
Now it's, I think, oh no, slides are not totally visible.
Just remove from.
Okay. I think it takes time, or yes, sir, your slides now visible, but again in the same issue, not in a full screen.
Now,
No, sir, it's just remains same. We can read your slide perfectly, I think.
but here I am.
So if the slide is moving, then we can read the slide properly. You can continue with the
You can continue.
presentation.
Go to the view and make it full
Yes,
screen.
sir. Yes, make
Yeah.
it. Yes, sir.
You see the view.
I make it in slideshow, but in my laptop it's showing its full screen.
Oh, in your laptop it is okay, no? So then you just start continue. So there is some technical issue.
The slide is moved. And it's,
Slides are not moving. We are in an introduction slide now.
okay, just give me 2 minutes.
Okay, surely, sir.
Now it is visible.
It's clearly visible now on the full screen.
I just created a PDF file that is it is high in size. That's it.
Okay, sir. Okay.
Okay, sorry for the inconvenience from my side.
It's
so-
absolutely okay, sir. Okay.
So we start from the beginning. So cannabis sativa belongs to the family Cannabaceae. It is an herbaceous plant, commonly known as Indian hemp. And in the previous literature, cannabis is mentioned as a three distinct species as cannabis sativa, cannabis indica, and cannabis ruderalis. But in the recent taxonomic research, considered the genus cannabis as a monotypic genus with only one species, that is cannabis sativa, and the rest of their previous species as a subspecies, that is cannabis sativa, subspecies sativa, cannabis sativa, subspecies indica, and cannabis sativa, subspecies ruderalis. The plant is native to the Central Asia, to the Southern Asia, but now cosmopolitan in distribution. And one of the native place is the Himalayan foothills of the India. So in Indian traditional system of medicine, cannabis is mentioned as a Vijaya or Bhang. There is several common name associated with this plant. There is one of the major controversial. The part specific common name was also mentioned. That is charas. That is the resin from the male and female flower. Ganja is especially the female inflorescence. Bhang, that is seeds and leaves. Its name. That is Atharva Veda named Bhang cannabis as a bhanga and used for the relief anxiety. Another one is Shushu Samhita. This is cannabis mentioned as a bhang, and there is used for the cataract and phlegm and diarrhea. In Ayurveda, the ingredients, it is used as one of the major ingredients for the pain relieving, stimulate appetite, aid digestion, and the several gastrointestinal disorders like nausea, dysentery, and diarrhea. And there is other uses, as we all know, they're used for the psychoactive effect like high in nature, recreational nature, and anxiety, depression, and insomnia management. So different parts of the cannabis used for the different application. This is fiber, essential oil and seed oil, hashish, resin, and phytocannabinoids. The plant possesses more than 500 specialized metabolites like phytocannabinoids, terpenes, flavonoids, etc. Till date, more than 110 cannabinoids was reported. Among them, cannabidiol and cannabidiolic acid, one of the important phytocannabinoids, because these two components is free from the legal restriction, and these two components are the non-psychoactive in nature. This is non-narcotic category. Whereas delta-9 tetrahydrocannabinol, that is THCA, and delta-9 tetrahydrocannabinol, cannabinic acid and cannabinol, that is THCA and THC, these two components are responsible for the intoxicating nature of the cannabis, and for these two compounds, cannabis is banned in several countries. But in recent research, overcome these two components and mainly focus on the CBD and CBDA for the development of the CBD genotypes or Semitic cultivar, which having low or very less amount of THC. There is reported pharmacological activities beyond the traditional uses. Cannabinoids significantly play important role in the neurodegenerative diseases like epilepsy, schizophrenia, Parkinson, Huntington, Alzheimer, etc. Along that, CBD also played a significant role and proven clinical efficiency in managing epilepsy, anxiety disorder, schizophrenia. And the CBD have the anti-inflammatory, anxiolytic, and neuroprotective properties. So if we talk about cannabis, the legal status is one of the major concerns associated with the plants. Cannabis sativa banned in the many countries due to the delta-9 THC, which is psychoactive in nature. In recent year, the research had overcome this ban in the many countries through CBD cultivar, which less than 0.2% in European countries and 0.3% in Asian countries. In India, still cultivation, possession, production, manufacturing, etc. is prohibited under the provision of the NDPS Act 1985. However, R&D work is possible with the permission from the state government and the central government. So to overcome this legal constraint, researcher was categorized cannabis into three distinct chemotypes based on the CBD, CBDA, THCA, and THC content. The first chemotype was TSC dominant, that is also known as drug type, where TSC is more than 0.5%. Another one is CBD dominant, where CBD is more than 0.5% and very less amount of TSC, that is less than 0.3%. And another one is balanced or intermediate type, where TSC and CBD percentage is almost equal and the ratio is almost 1. So the major challenges in cannabis research, especially in Indian scenario, the legal restriction, legal regulation, lack of awareness among the human being, morphological diversity is unexplored, legal limited studies, lack of researchers, high TSC content in the genotype found in the wild, legal compliance issues, differentiation between chemotype is not studied, and as a resulting limited CBD rich cultivar in India. So how we overcome this? So how should India approach the cultivation and research in cannabis sativa? There is three main aspects. This is low TSC cultivation. Another one is maintain strict regulation for economic and medicinal uses. And third one is develop CBD rich cultivar. Here we focus only on this, to develop the CBD rich cultivar. For this research, we take one mandate to find out the potential CBD genotype for selection and development superior individual for the future breeding purposes in cannabis sativa population in India, from India. So that overall study is divided into 3 parts: the pre-breeding, breeding, and the post-breeding. In pre-breeding activities, we focused on the broadening the genetic basis of the crops, as we already will see in the next slide. Then is breeding. This involves the direct selection and development of new crops through the pollination. And the post-breeding, these activities is related to the trait evaluation after the successful pollination, then seed production rate and the variety release, and the distribution of the newly developed varieties into the different parts of the India in different climatic condition and their trait evaluation. So first of all, pre-breeding. In pre-breeding experiment, we studied the collection of the cannabis sativa plant from the 5 North Indian states like Jammu Kashmir, Uttarakhand, Himachal Pradesh, Haryana, and Uttar Pradesh. These are the representative image of the collected plant. And we have collected the female inflorescence. This is most important aspect for the quantification of the CBD and TSC, and along with their seeds for the further cultivation in our institute in CSIR CMAB Lucknow. This is the field view. Here the nursery was raised in the open field condition, then seedling plant arises, then cannabis sativa then transplanted into the open field, main field, and more than 500 to 700 plant in each year we studied, and there is mature plant in the flowering stage was taken for the further studies. In nature, cannabis is represent as a 3 distinct morphology. First one is dioecious female. This plant possesses only female flowers. Dioecious male, this is only male flower. And another is monoecious, where both male flower and female flower present. As the proportion of the dioecious female is 51%, dioecious male is 41%, and monoecious is 8%, very less in nature. Then identification of the dioecious male plant. Dioecious male plant was identified by the presence of male inflorescence arising from the nodal region, and male flower is yellowish green in nature with very short in size, sepal ovate shaped, oblong anther, and very short filament was present, and numerous pollen grain was produced. Then identification of dioecious female plant. Female plant identified by the female inflorescence crowded in the apical leaf axil portion with female flower is mainly in green in color, then bract pubescent, lots of trichome present, and ovary globose, and feathery stigma with lots of papillae. The last photo is the same image of the stigma. Then architecture of the inflorescence of the monoecious cannabis sativa, where both male flower and female flower present in a single plant, and female flower is always above the male flower. However, the different morphological differences between male flower and female flower along with the dioecious plant is not very much. Then some morphological characterization also done in the field condition, that is plant height, stem diameter, leaf length, leaf width, internodal length, number of primary branches, number of inflorescence, herbage yield also taking for the trait evaluation. And then come to the micromorphological characterization of the pollen grain. This all character is important for the pre-breeding scenario. Here we all taking all the traits for the suitable traits for the further breeding. Here we taking the developmental of the male flower. The immature bud to mature flower takes almost 6 to 8 days and produce numerous flowers. Anther is 4 to 5 in number. Here, pollen is shown. The red coloration is viable, and 95% of the pollen viability was observed. And this image in the black background image is showing the pollen in vitro pollen germination, where two nuclei were shown. This is dinucleated in nature. One is male gamete, another is vegetative nuclei. Then structural analysis of the pollen grain was also studied. The pollen is isopolar, monad, spheroidal, with 3 aperture. There is 3 typorate, and there is a granular surface of the pollen is found in the scanning electron microscopes. And this granular nature is helped to the anchorage into the stigma surface. Then micromorphological characterization of the female flower. From the immature female flower to production of the mature seed takes almost 33 to 35 days, and numerous pollen grain were shredded onto the stigma and germinated, but only one pollen tube is entered to the ovule. This is the isolated ovule, and the red arrow designated the pollen tube entering into the ovule. And from pollen germination to entering into the ovule of the pollen tube takes almost 50 to 60 minutes approximately. Then micromorphological characterization of the seed. Here we found the total cannabis germplasm showing 4 different types of seed surface ornamentation. And there is coloration is brownish to blackish brown, and 4 different type of seed coat coloration. This is net-like structure, parallel black stripes, irregular black stripes, and black patches. This coloration is important for the camouflaging with the soil to avoid the bud predation during the dormancy period. Similarly, we also found one of the important features in the seed of the cannabis. There is presence of trichome onto the seed hilum region as well as the surface region. These trichomes help to anchor with the soil and retain the moisture during the germination of the seed. Then some of the quantitative traits associated with the seed morphology, this length, width, with ratio, 1000 seed weight, and basal caruncle. This traits is useful for the seed size and the seed germination, etc. Then come to the anatomical characterization of the stem. There is TS of the stem of the cannabis sativa. This also similar to the dicot stem, as already we all know. This is first of all is trichome in the outer epidermal surface, then single layer of epidermis, then 1 layer of hypodermis along with the collenchymatous layers, then fiber cell with lenticephorus duct, cortical cells, phloem, cambium layer, and vascular bundle are also visible, and pith region in the central. So we found one of the important marker traits for the cannabis stem outline. There's 2 types of stem outline was found. One is wavy stem, another one is circular stem. Wavy stem is responsible for the formation of the ridges and furrows, and the circular stem, though ridges are no ridges and furrows was present. In male plant, 92.33% of the wavy stem was found, whereas in female plant is 35.7%. This could be used as a marker trace in the vegetative stage of the male plants. So the ridges and furrows in the wavy stem is formation is due to the 5 to 7 layer of collenchymatous cell layer was formed in between the hypodermal region. This number of collenchymatous layers, collenchymatous cell diameter, and collenchymatous cell region thickness were significantly higher in the male plant, which produced the wavy nature of the stem. However, in circular stem, this is no presence of ridges and no formation of the colon-chymatous cell. Then come to the leaf anatomical study. There is no significant difference in the leaf anatomical study in the structure. However, the thickness and the size of the cell is different among the studied excisions. Very similar in the upper epidermis, along with one layer of palisade parenchyma, then spongy parenchyma, lower epidermis, and trichome in the lower epidermis, along with the midrib with presence of the vascular bundle, and the lots of parenchymatous cell with bulge out at the midrib position. Then come to the one of the major important, as we already told, the THC and CBD content in cannabis sativa is the main considering criteria for the cultivation and legalization of the cannabis. This we taking the female plant and male plant inflorescence and shade dried with the then powdered, then extraction with the help of HPLC. 6 different cannabinoids were studied, and these are the chromatogram of the THC dominant, CBD dominant, and the balanced intermediate type. And in nature or in North Indian cannabis population shows the higher almost 87% of the drug type plant that is THC dominant, or only 1.1% of the fiber type that is CBD dominant is found. It is almost very negligible in the Indian scenario. Then these are the 90 accessions of the total THC and total CBD content, and we found only one JK3, only one fiber type plant with very lesser amount, that is 0.25% of the THC, and this plant used as a parental line for the further breeding. We also studied some molecular characterization through the spot marker. Spot marker is nothing but a start codon associate marker. And this marker help to differentiate. These are the dendrogram. Here differentiated total plant into the two different groups. That is the CBD, very less amount of the CBD rich CBD content plant, and the all this upper group C1 is differentiated by the THC rich plant. Similarly, the population structure analysis also shown the red is the marked by THC rich genotypes, and whereas in green contain the CBD, whereas though both content the plant content both the THC and CBD is colored by the both blue, red and green coloration. Then come to the trichome diversity in the monoecious cannabis sativa plant. Cannabis sativa possesses 3 types of trichome, that is capitate sessile, capitate stalk, and the bulbous. And this trichome is important in cannabis research because the cannabis produce cannabis sativa. Cannabis produce the cannabinoids in within the plant, but however they store into the trichomes outside the plant. This red coloration in the capitate sessile, this is the histochemical test where we use the fast blue B solution with the TS of the leaf and the TS of the brake. And this color, the trichome presence of the cannabinoid turned into red coloration, and those designated the presence of the cannabinoids into the trichome. However, the bulbous trichome do not produce any cannabinoids, but they are produce the terpene. And the same scanning electron microscopy shows the density of the trichome, and here it shows the female flower having the highest amount of trichome density. And this also correlate with the phytochemical content, that is total CBD, total THC, and the total phytocannabinoids is the cumulative of CBD and THC is highest in the female flower, whereas the total phenolic content and total flavonoid content also higher in the female flower of the cannabis plant. Then come to the breeding aspect. In breeding aspect, here is studied this is the diagrammatic representation of the studies. Here we study only the pollinations. So as we already told, the identify the CBD accessions with very less amount of THC is less than 0.3%. Taken as a parental line, then collection of the seed from this plant, then nursery was raised, the transplantation to the field, then screening by the male plant, screening of the male plants through chemotypic marker, then removal of the THC rich male plant from the field, then cannabis quantification, cannabinoid quantification, that is THC and CBD, then only CBD plant taking for the seed collection. This cycle was repeated for the first year, second year, and third year, 3 years. And here we get the CBD genotypes. So we easily understandable by the next slides. And these are the transplantation. This is a diagrammatic image of the transplantation of the field in the vegetative stage, where no male or female flower express. In the next stage, we found the male flower arises, because in cannabis there is one of the interesting thing is male flower express in more than 120 days, and female flower takes more than 150 days, because there is 1 month of difference between the male flower expression and the female flower expression. The male flower express in the some of the plant, and here we taking male flower for the HPLC analysis, and the CBD and THC content was quantified, and here the CBD THC is marked as a CBD and THC. And then in the next step, we remove the THC rich male plant from the field and only kept the CBD rich male plants for the further pollination. Then female flower arises, and we taking the male CBD pollen from the male CBD plant and bulk, and also transfer to the female plants with the help of artificial pollination and cover the plant. These are the pollen crystal interaction. After pollination, we have done the have we done the success, the pollination is successful or not. We taking the more than 100 to 200 female flowers from different individuals, and in 15 minutes there is we show that there very less, that lower row shows the number of pollen. This is fine dots, fluorescence dot. This image through the fluorescence microscope. The pollen grain, very less amount of pollen grain, 1 to 2 pollen tube was found in the 15 minutes. Similar in the 30 minutes after successful pollination, 2 to 3 pollen was found. In 1 hour, this numerous pollen was found, but pollen tube is very short in during the germination, and 2 hours, 3 hours, 5 hours shows continuous higher number of pollen grain into the stigma, and lots of pollen tube germinated into the stigma. So here we confirm this, there is no self-incompatibility, and there are another mechanism which restrict the pollination with the recipient plant. Then come to the post breeding. After successful breeding, successful pollination, the we get the offspring, seeds of the offspring, and here we evaluate the traits of the offspring plant. So how we get the CBD genotype? In the natural population, among the 90 plants, we found only 1 plant with 0.2% THC content. In the first year, is more than 148 plant. We found 3 of the 3 offspring with very lesser amount of THC content, and CBD content is also very low. But however, in next generation, we found in the second year, number of plant is 466. Found more than we found 30 plants, 7 offspring, 8 offspring, and 30 offspring from 3 respective individuals. And in the third year, among the 509 plant, we found 71 offspring with 0.3%, with less than 0.3% THC content. So here is the overall percentage of the natural population along with the first year, second year, and third year. In first year, 87%, the drug type, the green portion showing the drug type, the THC dominant plant is approximately 87%, and in the end of the third year, it's nearly half of the overall population. Lower than the half of the overall population is 46% approx. However, the blue portion is in the natural population is the CBD dominant. It's 1.11%, but at the end of the third year, it is approximately 14% increased. Similarly, the balanced chemotype, that is intermediate type shows 12.22% in the natural population. However, at the end of the third year, is approximately 40%, and very interesting data associated with the intermediate I have shown you. In the cumulative content, here is quantify the THC and CBD content. The green box is designated the total THC content, and blue box is designated the CBD content. In natural population, the THC content is very high and CBD content is very low. However, in the first year, second year, third year, these both are overall in balance or approximately ratio of 1. However, in fiber type, there is fluctuation in the CBD content. The CBD is very unstable in nature, because CBD and CBDS conversion is going on. This year 1, year 2, and year 3. Year 2, we found the higher number or higher amount of CBD content. However, in intermediate type, we found interesting data that is in natural population that CBD content is low than the THC content. The green box is THC and blue box is CBD. But end of the third year, the CBD content is higher in the intermediate type, and in all the 3 cultivated area, THC is going to the low due to the pollination with the CBD-rich plants. And in drug type plant, yes, easily clearly visible, the content, the curve is decline, and the content of the THC is decreases from natural population to the year 1, year 2, and year 3. Then trait evaluation through the morphological characterization of same traits we already taken in the first, the plant height, stem diameter, leaf length, and these traits are useful for the plant breeding purposes, and their ANOVA analysis of variance ANOVA was studied for the statistical significance or not in the among the 3 years. Then similarly, the anatomical characterization based on the thickness of the epidermal layer, hypodermal layer, number of pollen-chimatous layer, pollen-chimatous cell thickness, fiber cell diameter, fiber region thickness, etc. were taking to through the analysis of variance statistical significance study. Then cannabinoid characterization, same, these all were studied based on the natural population and year 2, year 1, and year 3, and the analysis of variance and the ANOVA was studied for the most important in plant breeding, as BSc first year, second year students also studied the there is some plant breeding concept, that is coefficient of variation, critical difference among these all are important for the plant breeding purposes. Here the most importantly the heritability study, the broad sense heritability was studied in the 3-year study, where we showed that these traits were heritable or not to the parent to the offspring. There we found the leaf length, leaf width, and internodal length is showing the 77, 86, and 84, and another one is number of primary branches 83 are the % of the heritability character. This means the above the 70% of the 70% of the heritability is showing that these traits are highly heritable from the parent to offspring. And similarly, the total THC and total CBD and at the below of the list showing the 43 and 40 approx 40% approx. These traits is heritable, but these 2 traits is influenced by the environmental factors also. That's why they are showing the very less percentage in the heritability. Then come to the overall summary. In the present study, we showed the identified a significant chemical diversity in the 90 cannabis sativa accessions from the North Indian states, and majority are the drug type plant, approx 87%. A single fiber type plant, that is CBD dominant plant, made the government regulatory guideline with less than 0.3% THC content. Dioecious cannabis sativa showed significant morphological and anatomical variations that is very crucial for the breeding purposes. Notable diversity observed in pollen grain size, seed characteristics, stem, and the leaf anatomy. The wavy outline could be considered as a marker for the identification of the male plants. Chemical analysis revealed that breadth of the female flower contained higher amount of the phytocannabinoid, phenolics, and also the higher amount of trichome density. Over the 3 years of the selective breeding, the program removal of the THC plant from the field successfully increased the proportion of the CBD genotype from 0.11% to the 14%. The morphological anatomical analysis showed the significant variation in the CBD genotypes. Similarly, the cannabinoid profile revealed the substantial increasement of the CBDA concentration and the lowering the THC content. Detailed morphological anatomical characterization and selective breeding lead to the significant improvement of the desired traits and showcasing a potential for the producing commercially viable CBD plants. And this study is only a bricks to the foundation of the cannabis breedings. There is lots of work is remains, and this is all of my PhD work. And in my postdoc, I studied the abiotic risk factors in the 71. I found the 71 low THC plant. How abiotic factor is influencing the CBD and THC content and different abiotic factors. And then thank you. Thanks to organizing team for giving me opportunity to present my work here. And one of the major thanks given to Revenue Department, Minister of Finance for giving me license for my study, and my supervisor, Dr. Narendra Kumar from CSIR CMAB Lucknow, and my IIT Delhi supervisor. Thank you.
Thank you, Dr. Ghosh, for an eye-opening presentation. Probably that totally demystifies cannabis science policy with clarity. Thank you very, very much. And your expertise bridges traditional knowledge and modern science to explore the therapeutic and agricultural significance of cannabinoids. So thank you. And we already have such questions. So we are now open our question answer flow. So first questions are come from, questions are always for the first and second, for both presentations. Oh, okay. Here's the two first. First question is, how can CBD be used to benefit health?
I already showed that CBD itself is a very much beneficial for the treatment of the anti-inflammatory disease and neurodegenerative diseases like Parkinson, Alzheimer, etc. And in recent study in the last 2 to 3 years showed that in cancer patient, there is after chemotherapy, there is several nausea, loss of appetite, this kind of symptom was found, but cannabis overcome this type of symptom and the patient should relieve.
Okay. So the next question is, can we develop more plant types with high rate of CBD in future?
Okay. That's why this is the basic work, as due to the lack of licensing and lack of awareness, and there is strict regulation. Cannabis research is studied in the late 2017 approx. There is only 2 to 3 institute is CSIR CMAP, or I work, and CSIR NBRI and CSIR IIIM. We work in the plant breeding aspect first. The several clinical studies also done in the several NIPER and several institute of CSIR CDRI, this kind of institute. But for plant breeding aspect, only 3 institutes are working now. And this is the very basic work for the cannabis breeding in the future prospect.
Okay, thank you. And similarly, it's too brilliant. Okay, our next question is, some believe cannabis only affect mental health negatively. Are there conditions where it can actually help to improve mental wellness?
Actually, the content THC, the compound THC, this is one of the major cannabinoids that is responsible for the cannabis-related intoxication. But CBD is one of the more beneficial, and CBD is free from the any kind of legal actions. That's why we focus on the CBD, and CBD also helpful for the mental kind of disorders.
Okay, thank you. Now the next question comes from Nabidul Rana. What are the modern techniques used in breeding cannabis for medicine and industrial purposes?
There is several modern techniques that CRISPR-Cas9 also applicable. Applied further, there is 2 to 3 projects was ongoing in the CSIR with CRISPR-Cas9, where they can edit the THC region or responsible gene for the THC region. That's there. Several kind of advanced techniques was also studied.
Okay, thank you. For the next question comes from Vishwajit Mondal. What is the difference between THC and CBD? I think it's very basic.
Difference between actually CBD and THC are both a terpenophenolic type of compound, and both are isomer to each other. There is only one weight group difference among their chemical structure. That's why this is very much difficult to increase the CBD content. Both the content were synthesized from a single precursor molecule. That's why the environmental factor is very much responsible for increasing the CBD. However, we already found only 14% of the plant, where rest of the plant where THC is also present. That's why it is different to differentiate the two molecule based on this.
Okay. Our next question from Ankita Mohaldar. How do CBD and cannabis interfere with other medications? And what adverse effects can they cause?
Actually, this is not into my field. Actually, there's adverse effect. There is some study also ongoing. That's why this not interfere with each other. I don't think so. Other medications.
Okay, sir. Thank you. Now next, Shorab Roy asked that are traits like trichome density, plant height, or flowering time more influenced by genotype or environment?
Actually, both the factor is also responsible. Both the factor responsible for that genetics, also the environment, and that's why our next study is also on the environmental effect of the abiotic factors, how the environment influence these characters. This is ongoing studies.
Okay, sir. Okay, next comes from Kankana Ghosh. Are there any efforts to map the genome of indigenous Indian cannabis? How important this for breeding program?
For the precise breeding program, this program is also initiated through Government of India, and this is the ongoing study for the genome map.
Thank you. Ankita Mohaldar asked that our pharmaceuticals are that. I think there is something problem in the question, or oh no. So, CBD oil and other putting themselves in a large risk. Oh, okay. There's a full question: is are pharmaceuticals that sell CBD and other products putting themselves in a large risk?
No. There is government legalization was done. Some of the CBD oil, there is in market, in Indian market itself, there is several CBD oil product was found, and most of them are imported from the other countries. And it is there is lots of product on CBD oil is found in Indian market.
Okay. Next is how does cannabis affects the body? What is it? The endocrine system, I think.
There is two receptor in our brain. There's CB1 and CB2. So the THC is kinetically very fast and bind to the CB1, CB2 receptor, then CBT. So this two receptor is directly related to the endocannabinoid system and also regulate the endocrine systems.
Oh, okay. Now next from the Ankita Mohaldar, how will FDA approval of Epidiolex affect the scheduling of CBD and other cannabinoids products?
Sorry, ma'am, can you repeat?
Definitely. How will FDA approval of Epidiolex affect the scheduling of CBD and the other cannabis products?
Actually, in 2020, the Epidiolex is approved by the European FDA. It is CBD-based medication. Is implied for the children with Dravet system with high number of seizures. This medication is helped to reduce the number of seizures, and there are clinical studies is ongoing. There is another side effect on all these things. Studies are ongoing in the abroad.
Okay, thank you. Now next comes from Jashmi Devnath. How should I counsel patient on medical cannabis?
Actually, this work is for the people of India. That's why there is several work also come forward from cannabis. Then we should console the public to use the cannabis-based medication.
Okay, thank you. Next for Viprodash, what are the biggest misconceptions or myths people still have about medicinal cannabis in India?
There's only one myth: the cannabis don't have any medicinal importance. The cannabis is only for the intoxicating and several highness feeling and that kind of thing, recreational purposes. This is the major misconception.
Okay. Now next comes from Jasmeet Devnath. Again, he asked for what evidence exists to support the use of CBD oil.
There are several studies, anti-inflammatory, There is number of, in fact, there is anti-arthritic properties also there to reduce the inflammation in the joint pain, and also several studies also there. There's lots of studies in cannabis, CBD oil and CBD extracts.
Next comes from Otanu Shaduka. What are the specific challenges faced by Indian researchers in advising cannabis-related science?
Actually, major challenges is the getting the license for the research. This is the major challenges.
Okay, sir. The next question is: can we involve Indian farmer for the commercial production for cultivating certified medicinal cannabis?
Sorry, can you repeat?
Can we involve Indian farmers for the commercial production of cultivating certified medicinal cannabis?
There are more than 100 plus Indian pharmaceutical companies who worked on cannabis medication, but most of the cannabis, the raw material of the cannabis medicine is come from the abroad, and there is no cultivation permission is still in India. There is two state recent time, Uttar Pradesh, Uttarakhand and MP, just giving the government giving the license to the some of the companies for the cultivation or in a bounded manner.
Okay. Any participants have any questions? Please feel free to drop your questions. Participants, feel free to unmute yourself and use the chat box. There's another question from Ankita Mohalda. What is the future of medical cannabis? Right. What is the future of medical cannabis?
There is very important. As you search in Google as a future medicine of cannabis, there is abroad is doing very great. There is lots of countries who are studying and doing their research very well in cannabis. However, in India, due to the legalization issue, there is very less research, and actually there is no research in India. They are trying to start. That's why Indian research is very late in cannabis.
Thank you.
Should overcome in your future.
Definitely. There is another question from Autul Shaduka. How does the CBD contribute to climate resilience and ecosystem production?
I don't think so. I can't get the question.
Okay. There's a last question in our list from Deepanita Boshak. What are the side effects of CBD oil?
After certain dose, there is some intoxication issue also with the CBD. But there is toxicity in the certain limit that is not reported yet.
Right. Okay. Thank you, sir. Thank you, Dr. Ghosh. Thank you very much. We look forward for your just valuable insights from your work. Thank you very much. Okay, participants and all students, do you have any questions? Okay, now I kindly request our first speaker to take a few moments from the students if time and he permits. Hello, Dr. Roy. Am I
Yes.
audible?
Hello. Yes.
There's a few questions for us, sir. Can I?
Yeah, sure.
Okay. One minute, sir. Just. There's a question for Supriya Shokar. How long does it usually take for phycocyanin-based treatment to show results in clinical models?
Basically, that is not a well established for psychopharmac treatment. It's all under the process, and very few cases it's under doing the process actually. So we can't completely say how long it will take time to process the data.
Okay, sir. Thank you. There's another question from Vishwajit Mondol. He asked for, can phycobilisomes be engineered to carry or deliver specific drug to targeted tissues, such as tumors or inflammatory areas?
No. That is it's not possible because phycobilism is the complex system. So, and the receptor of the cell in the huge amount of size of the protein is not possible. So, but we can't use their component to how they actually use the medical science directly, because in cases different type of phycocyanin or the phycocyanin, they use the chromophore group, and each chromophore group is very much important for the medicine purposes. And in the definitely in the recombinantly we can define the structure, but it's quite impossible as a carrier of the phycobilism.
Okay. Thank you, sir. Thank you very much. Thank you. And participants, do you have any questions? Please feel free to unmute. Okay, now I think no, there is no such questions now. Okay, thank you very much, Dr. Ghosh and Dr. Roy. And
Thank you.
Thank you. And from phycobiliproteins as a medical therapeutics to cannabis biotechnology, I think both talk highlighted how modern biology is evolving beyond lab boundaries into real-world impacts. It's very engaging, and these insights are crucial for our young minds, researchers, and academicians to explore our interdisciplinary paths. So I express our heartfelt gratitude to yours. Sorry. Sorry, am I audible?
Yes, absolutely.
Okay, sir. Thank you. First and foremost, I sincerely thank our eminent speaker, Dr. Chitran and Dr. Dipan Ghosh, for taking the time to share with knowledge with us. Your insights were both enriching and inspiring. Thank you, sir. I extend special thanks to our patrons, Dr. Brojokishore Goswami and Dr. Subrata Roy, for their continuous encouragements, and appreciate the efforts of our IQAC coordinator, Dr. Dipankar Bhattacharya, and our organizing secretary, Ms. Shapna Tamang, HOD Department of Botany, for their detailed planning and support. And I also like to contribute to our co-conveners, Srimati Shangati Kundu and Mr. Rajkumar Sharma, for their valuable assistance in organizing in these events. And finally, my heartfelt thanks to all participants for making this webinar successful with your presence and enthusiasm. Thank you. Thank you to all. And now I request to our co-convener, Srimati Shangati Kundu. Shangati, am I audible to you?
Yes. Am I audible?
Yes, you're clearly audible now. They're requesting Shangati Kundu to take their vote of thanks to our eminent speakers.
Okay. Good morning, everyone. I, Shanghati Kundu, co-convener of this webinar, consider it a great honor and privilege to propose the vote of thanks on this occasion. First, I would like to express our deep gratitude to our invited honorable speakers, Dr. Chetan Roy, postdoctoral researcher of Pennsylvania State University, USA, and Dipan Ghosh, postdoctoral research associate of Center for Rural Development and Technology, IIT Delhi, whose insights and expertise have enriched our knowledge and perspective. We are grateful to our benefactors, especially to our Honorable President, Dr. Bojo Kishore Goswami, and our teacher in charge, Dr. Subroto Roy, for their immense support. I extend my sincere thanks again to our TIC Sir, Dr. Subroto Roy, and IQAC Coordinator, Dr. Dipankar Bhattacharya, for taking time out of your busy schedule to grace the webinar with your presence, and also thankful to Dr. Anirban Bhattacharya, convener of our seminar organizing committee, for their support and inspiration. A heartfelt thanks to our organizing secretary, Assistant Professor Shopna Tamang, convener Mrs. Shulagna Roy, and my other co-convener, Mr. Rajkumar Sharma, and all other members of Seminar Organizing Committee for their meticulous planning and smooth execution of today's seminar. I convey my special thanks to two supporting staff and the technical team who worked behind the scene to ensure everything ran flawlessly. I would also like to thank all the participants and attendants for your enthusiasm and engagement have been truly encouraging. Finally, thank you all once again and being here and making this event a grand success. We look forward to more such faithful and fruitful virtual summit and gathering in future. Thank you. Have a good day ahead.
Thank you, Shanghati Kundu. Thank you for your words. And our feedback link are already shared. And your feedback is essential for future academic events. Its certificates will be provided to all of the registered participants upon successful submission. Please let us stay connected through research, collaboration, and shared learning. Thank you once again for being a part of these academic celebrations. And with this, I would like to officially conclude our one-day international webinar on recent trends in biological science. Thank you to all. Thank you. And stay safe, stay curious, and keep exploring science. Thank you.
Thank you all for your active participation in today's seminar. We kindly request you to take a moment to fill out the feedback form. Your suggestion and valuable will help you improve future events. Thank you so much, everyone.
Thank you. Now we officially conclude.
Okay, thank you.
Once again, thank you to our eminent speakers, Dr. Roy and Dr. Ghosh. Thank you very much.